So we talked about what a nucleic acid is, which is the polymer, but there is also another piece of terminology that I want to clarify, which is the difference between a nucleotide, which is usually what we'll be talking about, or a nucleoside. These, again, sometimes are accidentally used interchangeably. Generally, they mean the same thing. When we're talking about these monomeric units, they are built up of a sugar, a pentose sugar. Remember, this means a 5-membered sugar, 5-carbon-membered sugar, and this nitrogenous base. This is the actual unique part of the nucleotide, the part that we usually recognize with it being described as an A, a T, a G, or a C. But also attached to the sugar, in many cases, is a phosphate group. And this phosphate group is there on a lot of those examples I just gave with small monomeric nucleotides that are used for energy, for example. And also they're utilized in the polymer building of our nucleic acids. Distinguishing between a nucleoside and a nucleotide just means that this phosphate group is there or is not there. Again, often, in almost all cases, we're dealing with the nucleotide, the structure that has the phosphate group attached. But there are some cases where you will see this nucleoside structure. So let's get a better look at what makes up a nucleotide and also some of the numbering that we use for nucleotides. So again, a nucleotide is built up on a pentose sugar. So it's good that we just went through our last chapter where we talked all about sugars. This is a 5-membered sugar, and maybe you can recognize it if you've memorized your sugars from last section, but this one in particular, its common name is ribose, which is one of the sugars that we will be seeing is utilized in these nucleotides. And you can see this is the basic cyclic structure of a sugar, and attached to its glycosidic carbon is where the base for our nucleotide, our nitrogenous base, will be attached. There are 2 types of nitrogenous bases found in our nucleotides, and they are generally classified as pyrimidines. These are, you can see, a 6-membered ring of carbons and nitrogens, or purines, which this is actually a ring made of 2 rings attached, a 6-membered with an additional 5-membered ring attached to it. So a pyrimidine or a purine type nitrogenous base is attached to our sugar by the glycosidic carbon. And then you'll see that a nucleotide also consists of a phosphate group, which here is attached to the 5th carbon on our pentose, where its OH group used to be on this sugar. We now have a phosphate addition. Now, when talking about places of attachment on a nucleotide, we use a numbering system for the ring that is the nitrogenous base. And you can see starting from this bottom nitrogen here and going clockwise around 1, 2, 3, 4, 5, 6. On a purine, you can see the nitrogen here is nitrogen number 1. And here we are actually going anti-clockwise, 1, 2, 3, 4, 5, 6, and the additional nitrogens and carbons 7, 8, 9 are numbered here. This is to talk about, if we wanted to talk about one of these areas having a group attached to it, we would say the third nitrogen or position 3 on the pyrimidine. Now, the numbering system that we're really going to focus on is not so much this numbering in the nitrogenous bases, but the numbering that we use for the sugar. The sugar portion of a nucleotide is numbered as well, and it's numbered using the same numbering system we saw when talking about carbohydrates, except we're using a prime after the number, and that's only just to distinguish the numbers that are in the nitrogenous base from the numbers that are in our actual sugar. And you can see, starting with the glycosidic carbon at number 1, this is 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. We're only numbering the carbons in our sugar. These positions are very important, and we will be talking about them often through the rest of the year. In fact, when talking about how DNA actually forms or how energy molecules are utilizing these positions, the positions along these, again, to stress, are important to know.